Welcome to part 6 of the DisProt curation training, Curation in DisProt, Thematic Datasets and Use Case. Here you can find the learning objectives of this part of the training, describe what the DisProt thematic datasets are, discuss the relevance of thematic datasets, explain the steps of IDP literature curation, interpret and discuss a DisProt use case, 82 protein. So let's see now what are the thematic datasets DisProts. These are collections focused on specific biological processes or organisms. They were introduced in December 2020 in DisProt, and they are created through collaborations with field experts. For examples, here we have cancer-related proteins dataset that was created in collaboration with Zsuzsanna Dostányi from Hungary. And here we have autophagy-related proteins, a dataset created in collaboration with Professor Vasilis Proponas in Cyprus. And also viral proteins dataset was created through collaboration with Philippe Lemercier from Viral Zone and Swiss Institute of Bioinformatics. Curators can contribute to dataset enrichment, so not only to new datasets, but also they can enrich existing datasets. Also, researchers have the option to propose the creation of new thematic datasets that can be discussed with the DisProt team. Upcoming new datasets in 2024 are RNA binding proteins and age-related disorder proteins. So, what is the process for the annotation and creation of these thematic datasets? We identify the dataset that we are going to create. We are going to select the proteins that are already in DisProt, that can be part of that dataset, and of course we are going to add new protein entries that are going to be part of that dataset. And we add tags for the dataset organization and enrichment. You can see here the VHL protein. There's two datasets associated, cancer-related proteins and autophagy-related proteins. The same way, FMR1 protein has two datasets associated neurodevelopmental disorder-related proteins and biological condensates. So the benefit of this thematic dataset is that we as a DisProt team and also our curators can focus also their curation efforts on specific areas of interest. And this also promotes collaboration and expertise in specific fields. But this also means that having these datasets created and available from the home page, it enhances the accessibility, the relevance of our curated information. So let's now use case of IDP literature curation overview. We have here this protein. It's available already in DisProt entry. It's ATG8 interacting protein 2, the gene is AT2, from Arabidopsis thaliana, and is part of the dataset autophagy-related proteins. So, first step is to identify the UniProt identifier, the UniProt session number, and the organism reported in the publication in order to create the corresponding entry. Then, of course, we want to add the information from our peer-reviewed article, and we are going to add our PubMed ID to the reference identifier field. We want to add the experimental techniques using the Ecoterm used for the identification of the disordered region or one of its aspects. So we will search in the Eco box our techniques. And we will specify the region boundaries and the structural state of the disordered region, and also of every other aspect that we want to annotate. We want to add the full entire sentence from the publication, one or more, if possible, that define this region as disordered. And we also want to say in which section of the peer-reviewed article we found this statement. So, for example, here we have a statement that comes from the results of the publication. If available, we will add MIADI information for further enrichment of the entry. So we have this peer-reviewed article about 82 protein. The transmembrane autophagy cargo receptors 81 and 82 interact with ATG8 through intrinsically disordered regions with distinct biophysical properties. So what we find out in this publication is the organism is Arabidopsis thaliana. The disordered proteins are indeed 81 and 82. Now we're going to analyze 82. The UniProt accession number of this protein for this organism is this one. So this UniProt accession number was used to create the protein entry in DisProt. The protein entry that is available here at this link. This is the identifier of DisProt. So what the authors do in the publication is the assessment of this disorder state of the N-terminal region of 82. They performed 5 experiments, including also size exclusion chromatography, circular dichroism, and NMR spectroscopy. And we created annotations for the disordered state, so using the IDPO term disorder. And adding also all the information about the region, the techniques, the data and statements extracted from the publication. Here you can find a summary of this information. So which is the publication, which is the organism, Arabidopsis thaliana, the protein, the UniProt accession number, the region boundaries, all the experiments performed by the authors to characterize the presence of disorder in this N-terminal region of 82, and the aspect we are annotating indeed, disorder. But this paper also assesses the functions of these disordered regions, because inside these disordered regions, N-terminal disordered region of 82, the authors identified the presence of an ATG8 interacting motif, AIM motif. This motif is located at position 14, 17, and these are the residues inside the ID region, 193. It has been annotated using the geotherm protein binding. The binding partner is indeed autophagy-related protein 8F, ATG8F, with this UniProt accession. But of course, an ATG8 interacting motif, an AIM motif, is also involved in selective autophagy that they study in the publication. So we also created annotations for selective autophagy. And here you can find the example of functional annotation for selective autophagy, which is a biological process term from gene ontology, for that specific ATG8 interacting motif. Here another summary of what the authors assessed in the publication. So with heat treatment, is2 hybrid assays, NMR spectroscopy, they identified that the region, including the NMR and terminal AIM motif, is at position 14, 17 inside the N-terminal region and is disordered, but also is disordered and it's a linear motif that is involved in protein binding and in selective autophagy. And finally, there are also some construct alterations that were added through MIADI, which are 2 substitutions in alanines. And this is the conclusion of the DisProt curation training. Thank you for attending the training.